Great. So, good morning, everyone, and welcome to our DNA extraction activity today. We are really excited to be here with you. A few things as we get started. If you want to do the activity with us, we encourage you to go ahead and grab some materials now. I've listed them on the screen. Just some water. You go ahead, go to any sink, about a quarter cup of water, a quarter cup of hand sanitizer. So I'm holding it up on my other screen. Or if you have rubbing alcohol, might be in your bathroom, about 91%. Or again, hand sanitizer works. Some salt, food coloring if you have it, but that's not necessary. Some cups and some dish soap. Probably a spoon to stir everything. So hopefully folks have materials. I'll give you a moment if you need to grab it. If you are ready, you can go ahead and answer our would you rather question, which is a pretty fun one, I think. Would you rather have a pet dragon or a pet dinosaur? You can type your answer into the chat box. I'm going to add my answer in there as well. I'm going to say, well, I'm seeing a lot of dragons again. The cheese stands alone. I'm team dinosaur. I love dinosaurs. I'm a big fan of Jurassic Park, but it looks like we have a lot of dragon fans. I get it. Dragons are pretty cool too. Once again, we're grabbing materials. Hopefully you have them. If not, that's all right. You can maybe take notes and do the activity at a different point. But we are going to move forward. And I wanted to share a little bit about ourselves. My name is Simone. I am an instructor with Science from Scientists. We are an organization that bring hands-on science to different schools across the peninsula. We work with 3rd through 8th graders. Really, really exciting. And now we are doing that virtually. This is one of our fun lessons that we get to do. And we also have Miss Esther, who is here. She will be responding to questions that you have in chat. So if you miss something, you have other questions, you can go ahead and type it into the chat and she can help you with that. Like she is writing some of the materials that you'll need in the chat there. I will also be asking you all a lot of questions out loud, but you're welcome again to respond in the chat or out loud. Some of the logistics, if you've been in school learning probably similar things to what your teacher has you do, we ask that you mute yourself unless you want to share something out loud, just in case there's stuff going on, there's sounds in the background that could be distracting or make it hard for us to hear. We love questions and we love to hear your ideas, so please do feel free to type in chat, or if you have a cool science fact you want to share, we love to hear those. I will do my best to keep an eye on the video screens, but if you want to share, you can try and raise your hand and I will try and spot you. But if I miss you, again, you can type into chat and Miss Esther will also try and keep an eye on those video windows as well. Can everybody give me a thumbs up if we can agree to these rules for the activity? Give me a thumbs up. Ooh, I'm seeing lots of great thumbs up. Excellent. Perfect. So, today, we are talking about DNA. I want to start by asking, what do you know about DNA? What are some ideas you have, things that you may have heard? You can go ahead and annotate on the screen, or you can type into chat. What are some ideas? Ooh, I'm seeing Sal. Yeah, Sal, if you want to unmute yourself and share, what do you know about DNA?
Well, DNA is the whole biological instructions for your body. If without DNA, we wouldn't be here.
That's
And
great.
DNA has all control rules for cell division. Also some like eye color, and some parts of your body are only controlled partially about your DNA. So I guess you could say that DNA is really important.
Definitely. I would agree. DNA is really important. You said some great things. You said it's instructions. Some things different parts of DNA code for different things. Really, really important. Thank you for sharing. I also saw Elizabeth had her hand up if you want to unmute yourself. Elizabeth. All right, let's move on to Sandy if you want to unmute yourself.
Like, DNA is what we're made up of. Everybody has a special pattern. It's different for everybody.
Yeah, that's great. So DNA is slightly different. That's definitely true. It makes us who we are as individuals. Yeah, these are great ideas. I'm seeing in the chat DNA is what we're made up of. Everybody has different DNA. Yes, we're made of cells. Inside of the cells, we find DNA. This is great. Do we want to add anything? We have a really good list so far. All right, I'm seeing Abhik. If you want to unmute yourself and share.
It's our genetic code, and it makes our traits like brown eyes or the color of our hair.
Yeah, definitely. Yeah, it codes for different traits. I have blue eyes and blonde hair, and so I have a certain sequence pattern in my DNA. Definitely true. Let's take maybe one more idea. Catherine, if you wanted to unmute yourself.
DNA is something that helps every different animal and plant and every different living thing to just be unique and be able to do something.
Yeah, definitely. Yeah, so living things have DNA and it's different and makes them unique. Excellent. I'm loving these ideas. Really great. So let's see if by the end of today, maybe we want to add any ideas to our pretty long list already. So if you look at this picture, this is a model, and scientists use models a lot to help us understand concepts. This is a model of a cell. Somebody mentioned that our bodies have lots and lots of cells, and inside of our cells, we find DNA. So you see there's kind of the outer shell, that's the outer part of the cell. We call it a cell membrane. And then inside there's a smaller ball, a smaller plastic clear ball that is, well, does anybody know where DNA is found inside of a cell? What part of the cell? It's kind of a tricky question. Does
The
anybody
nucleus?
happen to know?
The nucleus?
Aha, the nucleus. Excellent. We're great scientists today. So definitely DNA is found in the nucleus. And so the smaller clear ball represents the nucleus, but it's not just DNA. There's other stuff that's in there as well. You can see there's string and there's some pipe cleaners. So our activity today, if we want to extract, that means take DNA out of the cell, how do we do that? What are some things that we need to do first? Can I just reach in and grab it? Or are there some things we need to do first?
No, you can't reach in and grab your cells from your DNA because since your cells have a hard membrane, you just can't squeeze your hand in.
That's right. Yeah. So there's several layers between where the DNA is in the nucleus and the outside of the cell. And cells are pretty small, so I couldn't reach my hand into it. But if we wanted to do, what would be the first step if I have that ball? What would I need to do? Does my hand go through plastic? Am I a superhero?
No, but that would be pretty cool.
That would be pretty cool. Yeah.
If
Well,
I'm
so what?
correct, if I'm correct, cells use passive transport. They just sit and wait and have all the cells, oxygen, water flow through them. If I'm correct.
So our body likes to be in something that we call homeostasis, so a balance. So there are certain kind kind of, you can think of like gateways that let things in and out of different cells. That's pretty cool advanced science. So definitely keep learning science. You'll learn all about active and passive transport. Active meaning you need to do something, and passive means it's easier, just happens to reach that balance. Again, homeostasis is a kind of a fun, cool science word.
I believe plants are the ones that use active transport because they need to store molecules and keep them. And unlike our cells, which want to achieve homeostasis.
All right. And I love all the contributions, but I definitely want to make sure that all of our scientists get a chance to share. So let's see if we can practice raising our hand, and then I'll try and call on folks. And again, Miss Esther and I will do our best to make sure that we do call on different scientists and everybody that wants to share. So to get to the DNA, what do we need to do to that membrane, that first outer shell? I see Elizabeth's hand. Yeah.
It's you.
So, I think maybe you would need something to break the outer layer.
Yeah.
Because you can't just reach inside a shell, because first they're so tiny that your hand wouldn't fit, and also your hand can't go through solid stuff.
Yeah, that's great. And I saw in the chat also Ved had the same idea. So we need to break open our outer shell, that cell membrane, to get to the nucleus, right? And we see there's other pipe cleaners, other things inside of the cell we call organelles. We don't need to worry about those right now. And then our nucleus. Can my hand go through that hard plastic nucleus? What do I need to do to the nuclear membrane, we call it?
Break it.
Yeah, we need to break open the nuclear membrane so we can get inside. But then it's a little bit tough to see in the picture. The string and those pipe cleaners are all together, and I only want to get my DNA. So what might I need to do next? I don't want to take everything inside of the nucleus. I just want the DNA. Let's see. I see, oh, Sandy's hand. Yeah.
I think it's probably salt or something. I'm not sure. Because salt can create a lot of friction.
Okay. So I love that you're already thinking about what materials we might be using, also thinking about what we have next to us. So we might use different things for different steps, but we want to separate the DNA from what we call proteins inside of the nucleus. We want to separate DNA from all the other stuff inside of the nucleus because we just want the DNA. So again, to review, we have to break open that outer shell, the cell membrane. You have to break open the nuclear membrane, that inner shell, and then separate the DNA from all the other stuff that's inside of the nucleus. So we get to do this ourselves today. Are we ready to extract our very own DNA? Give me a thumbs up if we're ready.
I think I know why we brought hand sanitizer.
Oh, keep that thought in your mind because we'll review a little bit later. But I love that we're already thinking like scientists and why we have certain materials.
Oh, I think
so,
I know.
The first thing we're going to do is in one of your cups, I want you to pour about a quarter cup of water, like so. Actually, I am going to turn off my background so you can see me demo a little bit, not dinosaurs, a little bit better. So I have about this much water. So if you want to get some water in your cup, I'm seeing a lot of scientists hold up water. This is great, excellent. We are very prepared scientists today. I'm loving this.
I already have the materials next to me.
Perfect, I love it. Anyone?
My mom is going to bring me the dish soap later when I need to use it.
That's fine. Yep. So for step 1, does anybody want to read step 1 from the slide? What are we going to do?
Put your hand up. You know how you put your hand with reactions.
May I?
I don't know who is speaking.
Me, Salvador Lopez.
Sure, go ahead, Sal.
Step 1. Pour about 1/4 cup of water into a plastic cup. To the water, add a spoonful of salt and stir until it dissolves. The water should look clear.
All right, thank you very much, Sal. All right, so next we're going to take our salt, and you can just use a spoon, or I'm going to use a teaspoon. I'm going to pour about a teaspoon of salt into the teaspoon. Then I'm going to add it to my cup, and then I'm going to mix, mix, mix until it dissolves, and so the water is clear. So right now I can still see the salt, so I'm going to keep pouring, or keep stirring until all of it dissolves, meaning that water looks clear. So it might take a little while. Stirring, stirring, stirring. Still going. My salt is not dissolved yet. That's okay. Sometimes we need to be patient in science. Ooh, I'm seeing some stirring. Excellent. Mine's not ready yet. My salt has not dissolved. I can still see it. I don't have clear water quite yet. It will take a lot of stirring. Yep, it's kind of a good arm workout for your morning, huh? Especially me holding up the cup and stirring. Whew, could have really strong arm muscles after today. All right, I am almost there. Mine's getting pretty clear. Almost there. Depending on how much salt you have, you may have more or less stirring to do. All right. I am just about ready. So now my water is pretty clear. So my salt is dissolved. Oof. If you're still mixing, that's okay. I'm going to explain what we're going to do for the next step. Do I have a different scientist that wants to read step 2? Olivia, yeah, if you want to go ahead and unmute yourself.
Take a sip of salt water into your mouth and gargle it for a minute. Spit the salt water back into a fresh cup.
Yes, great. So I'm going to take my salt water. All right. And now I have my gargled salt water in a new cup. Oof, very salty. Reminds me of the ocean.
It was really salty, mine. It was like so salty.
Yes, we don't want to swallow it.
Tasted a bit like the ocean.
Yes, I agree. All right. Can my scientists hold up their cup of gargled water when you have it? Wow. Seeing some great gargled waters, some faces of people that have just had a lot of salt water. I had the same reaction. Great. All right. Go ahead and put your cups down. We're going to move to our next step. Do I have a new scientist that wants to read step 3? Kavi? You want to go ahead
Add 2 drops of dish to the cup with yourself in it and stir 10 times with
Step 3. Add 2 drops of dishwashing soap into the cup with your cells in and stir 10 times with a spoon.
All right, so now we've got our cup. Now we need our dish soap. I just pulled mine from my sink from my kitchen. So I'm gonna pour a few drops. Whoop, whoop. All right, not quite drops, but that's okay. I'm just gonna take a spoon and we wanna stir it. Mix that soap into your spit water. You don't want it to be too bubbly if possible, but you do want to get the soap, mix it around like so. All right. Hold up your spit plus soapy water cups once you've done that. Nice. I'm seeing lots of really great cups. All right, fabulous. Ooh, we are ready for step 4. Do I have a scientist that wants to read the first sentence in step 4? I see. I apologize if I'm mispronouncing this. Awak, awak, awak.
a few drops of food coloring. 2 1/4 cup of rubbing alcohol or hand sanitizer in a new cup. Slowly pour the hand sanitizer mixture into the cup with the salt, water, and DNA.
Fabulous. All right, so now we have our third fresh cup, and this is where we're going to take our hand sanitizer or our rubbing alcohol, and we're just going to do, I'm going to pour about a 1/4 cup, so I'm just going to keep squirting. Till we have about this much.
I don't really understand this step. Can you tell it to me again?
Sure. So first, we're just adding our hand sanitizer or our rubbing alcohol in our cup, about yay amount, this much.
Like 1/4?
Great,
Oh.
perfect. And then if you have it, you don't need it, but if you have it.
Are we going to pour it into a new cup?
Yep, so you have a fresh cup that you're adding your hand sanitizer or alcohol into. So this is our third cup, a fresh cup. And then if you have food coloring, again, you don't need it, but it does help. I'm gonna do maybe 1, 2, 3, 4, 5 drops.
Gentle.
Just so that it's this
5 drops.
really pretty kind of tangerine orange color.
3.
Again, it'll still work if you don't have food coloring, but all right, so we should have
I might have
2 cups.
I accidentally dropped in one extra food coloring drop. Is that okay?
That is perfectly okay. That's a cool thing about science is that sometimes, can I ask that maybe, yeah, we mute, perfect. Some things in science you have to be very, very specific and very precise. I remember in Miss Esther, we were talking about this, how we had organic chemistry labs that are really long and you have really specific steps that you have to follow, and other things that you don't have to be as careful and precise. And this is one of those things where you don't have to be as precise, which is kind of nice. All right, so that second step, we are going to pour our food coloring and hand sanitizer or alcohol into our spit and soap cup, like so.
Are we supposed to mix up the hand sanitizer and food coloring before pouring, or just pour it like it is?
Yep, mix it up.
Okay.
So now I'm gonna mix. All right, and now, do I have a scientist that wants to read our step 5? Maybe a scientist we haven't heard from. A new scientist voice. Somebody want to read? I am using hand sanitizer. Let's see, do I have any new scientists that want to read? Oh, we're just so engaged in this step. I get it. This is really fun. All right, any scientists? Ellie, if you want to go ahead and read.
Wait a few moments, then hold your cup to the light and view the white clumps of DNA. They'll be on the top first, then sink to the bottom.
Aha, thank you. So if you can see, it's kind of tricky in this picture. This stuff, the white strands, that is your DNA. So sometimes you have to wait a little bit. Sometimes it depends on the temperature. I know it's a little colder out today. It works better if it's cold. But you want to wait and then hold it up to the light to see if you see any white bits in your cup. Wow.
Raise your hand.
It works. The experiment works. I was able to spot the white clumps up in sunlight.
Yes. Ooh, I'm seeing so many scientists were successful in their procedures.
I was successful.
Nice. Hopefully we're starting to see our DNA, some white clumps.
I saw 2 pieces of DNA.
Whoa, that's awesome.
I saw more than 5.
Yeah, I'm seeing some, I don't know if it'll translate. I don't think you can see it, but it does. Mine looks stringy, a little bit like snot. It's okay if you're still waiting for some of your DNA to separate out, but it looks like we've had a lot of success already. So I'd love to hear, or if scientists, if you want to hold up your cup to the camera so we can see. Ooh, very cool. Nice. Looks like we did a great job with our DNA extraction. I'm loving all the different colors I'm seeing as well, some blues and pinks. Ooh, purples. Give me a thumbs up if you're able to see some of your DNA. Whoa, so many scientists were successful. Again, it's okay if you haven't seen it yet. Temperature, again, is a trick. So if you didn't see it now, you can try putting it in the fridge, the refrigerator for a little bit, and then checking on it in a few minutes. So then you should be able to see some of your DNA. But for those scientists that were able to extract, that means take out some of the DNA, I'd love to hear, what does that look like to you? How would you describe it? What are some great observations you can make about your DNA? Very cool. Alex, you want to go ahead and unmute yourself?
It looks like snot.
I agree. I think it does look like snot. Great. Anna, if you want to unmute yourself, how would you describe your DNA? Yeah, I'm seeing some consensus. How about Meg? Meg, did you want to unmute yourself?
They kind of look like different sort of strands. Mine's a bit like big and flat. A bit, yeah.
Mm-hmm. Yeah. Okay, so big, white, snotty, excuse me, stringy. Yeah, Meg, if we wanted to go back, I don't think that we got to hear from you. I see your icon there.
Yeah. Hey, I see mine.
She can't hear you.
Yep. So how would you describe, what does your DNA look like?
Say it again, Sage. I don't think she heard you.
My DNA, it looks a bit like flat string. It's basically just like, really stringy and a bit cloudy, yes.
Okay, cloudy. Yeah,
I have a question.
great. Sure.
Oh, how come my DNA when I put how come my DNA floats for a little while, then sinks instead of sinking like the second it goes?
That's actually a really good question. I don't know the answer to that. Miss Esther, do you happen to know why it floats and then sinks?
I think it has to do with the density or how much stuff there is in the amount of space of your alcohol, your water, and the mixture of the two. So when you first put the alcohol and the water together, the alcohol sort of floats on top and it has a different density than the water. And the DNA floats in that. It's less dense than the alcohol. But then when the two have a longer time together and they mix together, the density of that mixture is the DNA has a greater density than that mixture, so it sinks at that point. So it's actually pretty cool chemistry example of the effect of density on mixtures.
Great, thank you.
Thank you.
So, one question, I know we have some similar observations in terms of how we describe our DNA. Do we think we're looking at one single strand of DNA or one small part of our DNA?
No. Our DNA is
shaking.
well over 6 feet long. And that's just one.
So we have a lot of DNA in our bodies. And so what we're seeing, those stringy bits, is actually lots and lots of DNA all clumped together. We couldn't see just with our eyes a single piece of DNA. It's way too small. Because think about how many cells, we can't even see our cells mostly on our skin or other parts of our body. And then the DNA fits in one small part of our cell. So it's really small. So what we're seeing is lots and lots of DNA all clumped together. And it's your DNA. It's DNA from your body that's unique to you, which is pretty cool. So oftentimes you do fun things in science, but I always think it's important to think about why we do it. So I'd love to ask you all, why do you think we want to extract DNA? What could we learn, or what kinds of scientists might want to extract DNA, meaning take DNA out of cells? Why do we want to do this process? Elizabeth, yep.
I think that maybe doctors would like to do that because they could see more how DNA works and stuff.
Mm-hmm. Yeah, I love that. So we want to see what it looks like. We want to learn more about it, and we can learn maybe more about ourselves. That's great. So maybe we can understand diseases better, and so we can help people stay healthy and treat different diseases. So people that work, say, in hospitals or do research around human health definitely might extract DNA. Do we have other ideas? Olivia.
So like people can use DNA to use in crime scenes to figure out the suspect.
Yeah, that's great. So we know we have DNA in our cells. So if somebody goes and they rob a bank, they'll probably going to leave some of their cells around, maybe from their hair, from their skin. And so then different kinds of investigators, forensic scientists can go and figure out who was there based on the DNA they left behind. And they can match it because DNA is unique to the person. And so they can figure out who exactly was there, and they might be a prime suspect for that crime. So definitely forensic scientists. There's all those TV shows that you may have seen, or movies, where they use really cool science and DNA to figure out who committed what crime. And I also wanted Miss Esther to share a little bit because she is a scientist that studies not humans, but birds. And so she got to use DNA. So I wanted to give her a chance to explain about how she used DNA.
All right, so I'm interested in animals that live in social groups. So they live in family groups, kind of like we have family groups, except their groups are bigger. So they have a bunch of possible moms and a bunch of possible dads, and they all together raise one group of kids. The particular species I studied is a bird, but lots of different animals do this, mammals and birds. I don't think we found a reptile that does it yet, but I'd be really interested if we do. Sometimes insects have this kind of family structure too. And when you have a group like this, you can't tell whose kids are whose, uh, at least not by looking. I can't. So I spent a lot of time going around and collecting a little tiny blood sample from all the animals that I was interested in studying. So I would go out and I would catch them, which took a lot of work. Catching birds is not easy. And I take a small blood sample from their arm or their wing, just like you would get a blood sample taken at the doctor's office, very small sample. And then I was able to take that and take the DNA out of that blood sample. Bird's blood is different than ours. It's got more DNA in it than ours does for cool reasons. So they have more DNA in their blood. So I got a lot of DNA out of the little tiny blood sample. And then I could ask the question, whose kids are these? And figure out for my birds, which of them were actually having kids in the group, which ones were not having kids, but just helping out. And so I was able to get some information from them that I wouldn't have been able to get otherwise. And so I got pictures that are kind of like this. It is a little bit rude if you think about it. Like if you were walking down the street and somebody suddenly threw a net over you and then took a blood sample, you'd probably be upset too. So they were definitely mad at me, but the birds that I study are about this big. So they like raise all the feathers on their head and they glare at you. And it's really cute because they're this big and they sound like rubber duckies. They have a call that goes me, me. So they're very angry, but they're really cute about it. So I would extract the DNA and then I would sequence or figure out the actual code, the DNA code of a small portion of it. And I would then compare different portions. Let me turn off my virtual background so I can show this to you. This is just a drawing that sort of represents what I would do, but I did this in a computer program to make it more accurate. But I would extract the DNA and I would get little peaks or base pairs in certain positions. So it looks something like this in my computer program. And so here I would have this individual's DNA from the group and this individual, and then I'd have this kid. And so if I look at the DNA, and you guys can look too, do you think the top one or the middle one is more likely to be the dad for this kid? What do we think? The top one or the middle one?
Middle.
Yeah, the middle one. And why do we think the middle one is more likely the parent, the dad for this particular kid? Why do we think so?
It matches better.
Exactly.
More similar.
It's more similar, yeah. So our
there a
DNA, we
spotlight?
know we get our DNA from our mom and our dad. And so by looking at all the possible moms and dads and then the DNA that the kid had, I could figure out which of those possible parents the kid actually belonged to. So I did this for my birds and it helped me figure out why they lived in groups by studying who was actually having success reproducing. It helped me understand why they lived in groups and what it was about their environment made them live in groups. Because we know most birds don't live in groups, so there has to be something special going on for them to have to live in groups. So DNA is actually a really important part of some of ecology or understanding why animals do what they do, understanding family structure of animals. So that's a little bit about my work and why I think DNA is super awesome.
Thank you for sharing. Did anybody have any questions for Esther? Esther, I can say, is one of the best people to go walking outside with because she always has a great eye for spotting birds or other cool animals. So I always learn a lot from spending time with Esther.
It looks like Sal has a question.
Yeah, Sal.
Two things. First, is this like the same thing as Ancestry.com?
It's similar to Ancestry.com. Ancestry.com, I think, probably uses single nucleotide base pairs instead of microsats. So slightly different science process, but similar idea, comparing DNA between individuals to understand where the ancestry came from. I think I'm not entirely sure about what process Ancestry.com and similar companies use to compare DNA. I think they might have to use not only the DNA in your own DNA, but also the DNA of your mitochondria, which are inside your cells. They're single organelles that help us turn food energy into stuff our cell can use. And every cell has them, and we get them from our mom entirely because our mom has the cell that sort of begins us. And so that is where our mitochondria comes from. So they might use a combination of our nuclear DNA or the DNA inside of our nucleus and the DNA inside of our mitochondria to trace our origins, because you can trace back better in mitochondria DNA because you know it has to come from mom because of the way that people are formed when they're first, when the cell first gets together and you start as a fertilized egg.
Awesome.
And can I say my second thing?
Sure.
And if I'm correct, the bases of DNA are cytosine, guanine, adenine, and thymine. If I'm correct.
Mm-hmm. Yep, that's the code of DNA. Your DNA is a bunch of those bases in different orders, and it tells the cell how to build different things depending on the order of those bases.
And I noticed a lot of scientists were drawing on the screen the shape of DNA that we call a double helix because it's two strands wrapped around. So I love that we're sharing all these ideas that we have about DNA. So I wanted to go back to this idea about a procedure and how we did things. Again, it didn't have to be very precise, but we did have to do certain steps in order to successfully get DNA out of our cells. So I want to think about why we use the materials that we did. So the first thing we did was gargle with salt water. So thinking about, why do you think we needed to gargle with salt water? Why did we gargle with salt water?
DNA in your throat.
Yeah, so we needed the salt, helped scrape the cells off of the inside of our cheeks. And without cells, we would have no DNA. We just have water. So we needed to gargle with salt water to scrape the salt, the hard edges of the salt, scraped it off of the inside of our cheeks, and then we spit it into our cup. How about the soap? Why do you think we wanted to add the soap? What did we do with our cell membrane? What did the soap do? Yeah, I'm seeing people point to it. That's great. What do we think? What do we need to do? What was our first step? We needed to break open our cell membrane.
Yeah, break it open, break it open so
Yeah.
we can see the DNA.
Definitely, we needed to break open the cell membrane. And then we had, any guesses, why did we use, what is the word? Food coloring. Again, not necessary, but how might this help us in the process? It just basically helped us see a little bit better, because if you had white DNA and kind of a clear liquid, not really easy to see with your eyes. And so white shows it better against a colored background. So it just helped us spot the DNA. And how about whether you used hand sanitizer or rubbing alcohol? Why did we need that? Any guesses?
I think I know why.
Sure.
The hand sanitizer help with the DNA help. No, not DNA. Food coloring help make it easier to spot your DNA.
Yep, so we added the food coloring, it helped us spot it, and the alcohol helped us precipitate, meaning separate out the DNA from other stuff, and so it's separated from the mixture, because we just wanted to see the white, snotty, cloudy looking clumps of DNA, and so we needed to add the alcohol to help it separate from the soapy mixture, soapy, salty mixture. So again, pretty basic procedure. You could do this again, and you could try this with different types of cells. You could do it with, it works well with strawberries. So instead of gargling with salt water, you can mash strawberries. You could do it with different kinds of fruit. You could do it maybe try and scrape like cells from your arm. You can do it, compare yours with somebody else that lives in your home, maybe a sibling, a parent, which is pretty fun that you can just do this over and over as long as you have the materials. So now that we know a little bit about how we extract DNA, we're going to kind of zoom out and think big picture, right? A lot of scientists mention that DNA makes us unique, but I want us to think about just how unique. So we have some quiz questions. So you can go ahead and type your answer into chat. This first question, how similar are we to the person next to us? Right, so how similar are you to the person sitting next to you, or even to me? How similar are you to me? You can go ahead and type your answer into chat. How similar do you think we are to other people? We look very different, perhaps. And DNA accounts for those differences, partially. All right, I'm seeing a lot of A's. Do we think we're half similar to people? All right, okay, a little bit of a mixed bag. So maybe we think that we're a little more similar to family members. Are we ready for the answer? We are 99.9 similar. Our DNA is practically the same, but that 0.1 difference accounts for all the differences in the way that we are, which is pretty cool. Yeah, mind blowing. We are all 99. Everybody in this meeting, we are 99.9. Our DNA is 99.9% similar to everybody else in this meeting right now.
Does anybody
Super
know
cool.
or have a guess why we're so similar to everybody else? What do we think about our DNA makes so much of it the same from person to person? What do we think?
Alex has his hand raised.
I think that we're 99.9% the same, because if we're not that close together with DNA, we can't have babies.
Yeah, so being able to recognize each other as the same species, the shape of our DNA is the same. Yeah, but the pattern is 99.9% the same too. So think about it. Humans have a lot of things in common with other humans. So think about, how about eyes? Does everybody have eyes? Yeah, almost everybody. Or a heart. Everybody got a heart? Our heart's pretty similar from person to person. Yeah. What about our lungs? Are our lungs pretty similar from person to person? Do we all have hands? Yeah, most of us. We all hopefully start with them. So all of the DNA that tells your body how to build hands, how to build your eyes, how to build your heart or your lungs or your digestive system, all the inside stuff that makes you work. Yeah, ears. Yeah, all that stuff, all that code is the same from you to everybody else. So all the stuff that makes your body actually work as a human being is the same from person to person. All that DNA takes a lot of information to build eyeballs. It takes a lot of information to build a brain. It takes a lot of information to build your heart too. So all that stuff is the same from person to person to person. And in 0.1% of your DNA is all the other stuff that makes you look different from other people and feel different and think about things differently. So a lot of the stuff that makes you human is the same from person to person, 99.9%.
Excellent segue. Thank you, Esther. Super, super fascinating. So taking that understanding, how similar do you think we are to chimpanzees? Go ahead and type your answer into chat. Is our DNA 90% the same, 80%, 98, or 20? Well, a lot of C's. Wow, we're very enthusiastic about our answer choices. It is indeed C. We are 98% similar genetically to chimpanzees. Pretty fascinating. I know somebody mentioned earlier about evolution, which is another way that scientists use and try and understand DNA so we understand tree of life. So evolutionary relationships, who's related to who, which is pretty cool. All right, how about a banana? How similar are we to bananas? What do you think? Okay, looks like a little variation. We are 50%. Our DNA is 50% similar to the DNA of a banana, which is pretty silly. I actually have one next to me. So we're half the same as this banana here, which is pretty wild. And again, we think about that because there's certain similarities in terms of what are inside of cells of chimpanzees or even bananas. And so the same code for DNA codes for cell, well, they have cell walls, different parts of the cell, different structures inside of the cells. And so we have some similarities in our DNA. All right, how about this? Which organism do you think we're the closest to? Who are we most similar to? Oh, yeah, we are most similar to cats, right? We have, I had a cat growing up. Again, look very similar, or doesn't look very similar to humans. And any guesses how similar? What percentage of our DNA do we share with cats? Any guesses? 75,
95, 95,
100, 95,
100.
90. We're 90% similar to cats. So, yeah, pretty surprising when you learn some of these things. Again, on a genetic level, our DNA is 90% similar.
Is D a mouse?
Mm-hmm.
Oh, I thought. Wait a second. I just want to check. I thought we were closer to mice than cats. But hey, I like cats better than mice, so go cats!
We'll see how similar we are in just a moment. So I love the segue.
I love cats. They're my favorite animal.
Same
I love
over
them
here.
too.
Same
and
over
my
here.
and my favorite kind of cat is a tiger.
Well, the tiger is in the feline family, so technically the tiger is a cat, except it's just not a house cat.
All right, scientists, which organism are we farthest from? If we can go ahead and mute for now. Which are we farthest from? Looks like we have some pretty astute, pretty observant scientists. It is indeed C, the fly. And we can see some of those percentages. We are 61% similar, 80% similar to a cow, and about 85% similar to the mouse down there. So fairly close, 85 and 90. Pretty impressive. So I know we are running out of time, scientists. We just have a few minutes, and I wanted to see if there are any final questions that came up. We did a fantastic job extracting DNA from our mouths. We got to look at what our DNA looks like and learn a little bit more about why we do this, why it's important.
I have a question. You taught us how to extract it, but how do we actually, can we take it out of the jar?
You can take it out of the jar and put it maybe like in a little plastic container. It'll last longer if you put it in the fridge, and it will break down over time. But if you want to show it to somebody later today, yeah, you can take your DNA and keep it in a closed, sealable container.
Cool.
And again, I recommend keeping it in the fridge too. Any other questions that came up, scientists?
Wait, would it work if you put it inside the freezer instead of the fridge?
Yeah, so that's where oftentimes if you're using this for research purposes, they keep them in giant freezers and they have lots of DNA samples so that it stays over time.
Cool.
Just don't forget about it.
Yeah, you don't want to spill that in your fridge. Oh.
That's yeah. All right, scientists. We are just about out of time. Hopefully you've had a great festival so far. Hopefully you have other fun things to do. You did a great job learning. I'm very impressed with all of the knowledge that we shared. I learned a lot from you all as well. So Esther and I really wanted to thank you again.
See you guys.
And
If you attend the next one, then you can see.
Bye, everybody.
Bye, everybody.
Bye.
Thank you for hosting this Bay Area Science Fair. I really like it.
Thank you.
thank you.
Love it. It's
In future things, which are like, I don't know, a couple hours away. Bye.
Bye.
Bye, everybody.
Bye.
Bye. Bye.
Bye.
Oh, thank you for hosting these, and whoever had the idea of making these, please thank them. Please thank them for thinking of these, and thank you for hosting this.
Yeah, thank you for joining, Sal. I loved all of your ideas. You were great today.
Thank you.
Have a great rest of your day.
You too. If you're gonna be hosting any more, I'm gonna join in a couple of hours.
Oh, nice.
So keep an eye out for this tag. Sal